Hi everyone, my name is Zihan Yan. I'm now a junior studying bioengineering, and my concentration is cell and tissue. I'm also an undergraduate researcher at Shiver Lab. I'm currently working on a project of Huntington disease mouse models under the guidance of Dr. Simpashu and McCormick for almost 3 semesters. It is therefore my greatest pleasure to be able to share some of our previous and also most recent research progress for you all for the virtual cow day. Huntington disease is an inherited, progressive neurological disorder characterized by degenerating striatal medium spiny neurons. The cell replacement therapy proves to be an effective way to treat the disease. To facilitate clinical translation, the overall aim of our research is to test the efficacy of striatal progenitors that were generated from human pluripotent stem cells within the fully defined and scalable 3D hydrogel. To test the efficacy, we did intracranial injection of progenitor cells into HD mouse models. The two clusters of progenitor cells that we tested on, or MSNs and OPCs, were set up different experimental conditions to test different cell combos, and the responses we would like to measure were listed on the bottom left on the slide. So first, I will briefly talk about our stage 1 findings on the survival analysis of HD mouse models that was done in the last semester. Based on the plots we got, we summarized that MSNs and OPCs significantly extended lifespan in HD mice models. MSN plus OPC combo did not show significant improvement compared to MSNs or OPCs alone. Addition of hydrogel did not significantly affect the survival either. Then I'll explain and talk more about the progress of immunochemistry that was done this semester. After immunostaining and mounting of the brain samples, I looked them under the Zeiss microscopy to first identify potential image of grafts under different combos of antibodies. Then those samples were brought to the Opera Phoenix for higher resolution screening and image analysis. Starting from the HD mouse models, we identified 2 sets of grafts, one from H12 and one from H5, that were kind of interesting to us. The cell combo for H12 is OPCs and MSNs in suspension, and the brains were harvested 20 days post-implantation. The markers we use to signify the existence of grafts is basically HNA, which is also an immunostain for human nuclei. From both the top left picture and picture on the second row, we can see the survival of HNA-positive cells 20 days post-implantation in the striatum. Similarly, still for H12, we change to another set of immunocombos and get this picture, where there are positive signals for HNCAM, a marker of human neurons. On the contrary, for H5 cages, the experimental condition of which was OPCs in the gel and was harvested 2 months post-implantation, HNA signal doesn't co-localize with DAPI. Therefore, one reason could be the non-specific binding from blood vessels, for example, or due to dying cells as a consequence of the progression of the disease. In another direction, to better understand immunological factors underlying survival and rejection of intracranial neuron transplants, we also harvested brain samples from wild-type mice that had different CSA administration conditions post the surgery. CSA is typically administered as a standard protocol for the purpose of immunosuppression after the surgery. For those with CSA withdrawal condition, the 1 month post-implantation with CSA, followed by 1 month without CSA, we identified some image of grafts from them using the marker HNA. In the same set of brains and similar location of the grafts, we discovered PD-1 signal, which signifies the existence of immunocells and immunological responses. We also discovered image of grafts in both CSA positive and CSA negative situation. CSA positive means that the wild types were injected with daily CSA for 2 months post-implantation, and CSA negative means that the wild types were not injected daily CSA. In conclusion, we summarize that MSNs and OPCs survive for 20 days post-implantation in HD mice. However, they fail to survive for 2 months post-implantation. This result comes from H5. From the immunotests, we see that CSA-negative condition gave us one of the best HNA stainings we have found so far. It shows high viability suspension injection and has perfect nuclear, and the cells look healthy and even migrating outside the injection site into the striatum. This result is interesting in 2 ways. On one hand, it indicates that CSA doesn't seem to be necessary in wild-type mice to guarantee survival of human cells in the striatum for 2 months. On the other hand, comparing this result to the ones from H5, we can see that the most likely cause for why we couldn't find a healthy graft in H5 is because of the fast neurodegeneration in HD mice that affected the survival of implanted cells. So this is the end of my presentation, and thank you all for your listening.